Hello, everyone, and welcome to the second episode of ALS Clinical Trials Unboxed, brought to you by ALS Canada. It is a huge pleasure to have our special guest here today. She is the co-founder and VP of Research and Development for Progenics. Maybe you will also know that for many years she was an associate director at Project ALS and is here to talk to us about PRO 101 program, also known as Prosetin, and how that's developing with also a Canadian site involved at this point, and just all of the work that's gone into really developing something from a very ALS-centric standpoint through to clinical development. So we're so pleased to have a special guest here today, Erin Fleming. Thank you for being here, Erin. We're going to give you the floor. You have a few, about 15 minutes for presentation, and then we're going to have some time for some questions. So thank you, and the floor is yours.
Thank you, David. Thanks so much for inviting me to present on this series, because it's excellent timing. We're just about to launch the first ever study of Prosetin for people with ALS, and there is indeed a Canadian site. So I know this is a quick presentation. I do want to give a little bit of background on why Progenics and Prosetin ever came to be, and then I'll try to reserve some time at the end to talk about the details of study PRO 101. So really quickly, Progenics was founded about 2 years ago, and it was started by Project ALS, the nonprofit, Dave, that you mentioned, researchers at Columbia University who had been working with Project ALS for close to 25 years on developing new models for ALS and then using these for drug screening, and a venture capital fund called Medical Excellence Capital. And it was really started with the explicit mission to develop Prosetin for ALS. And hopefully we'll do more than that over time. But we were very excited about Prosetin and wanted to make sure that we had the funding and infrastructure to move this promising compound from academic research into clinical trial. And Prosetin, I'll talk about this in more detail, but at the high level, it is a novel drug. It is a potent MAP4 kinase inhibitor. I'll talk about the mechanism a little bit more also. It was designed by our co-founders at Columbia. It's an oral drug. It's actually administered in a liquid solution, and it's highly brain penetrant. And we've seen in a range of preclinical studies that Prosetin can rescue motor neurons derived from people with ALS against endoplasmic reticulum stress and other core disease stressors. And study PRO 101 is the phase 1 study, so the first in human study of Prosetin. We started with very quick sections of the study in healthy volunteers just to test single and then a bit longer periods of dosing to make sure that it was behaving as expected. There were no safety issues. And now we are about to start part C, which is the first evaluation of Prosetin for ALS, again, starting this summer. Really quickly, Progenics is a very small team. And as you mentioned, we came out of a community effort. So we have a very clear mission. I am a co-founder. Our other full-time employee, Stan Abel, is our president and CEO and has a really good experience, a lot of experience running small biotechs from a business perspective and steering the ship. Our inventors and co-founders at Columbia and Project ALS are also still really deeply involved in the program. And I think equally important, all of our decisions are guided by an amazing community advisory board and scientific advisory board to make sure that these decisions for our clinical program are being made in an ethical, humane, and scientifically rigorous way. So to take a step back, again, to the question of why we are even here in the first place, why Progenics exists. At the very bottom line, our approach is rooted in this basic question that Project ALS asked 25 years ago, which is a basic problem, I think, for ALS drug development. If we don't know how motor neurons work normally, if we don't have good models of motor neurons, the brain cells affected in ALS, we have no chance at really understanding what is driving their demise in ALS. And intervening in a rational way. So one of the first things that Project ALS funded was an effort to create better models of ALS, to actually be able to study these in a laboratory setting. And they actually funded one of our scientific co-founders at Columbia, Henrik Victorly, in 2000 with this sort of blue sky idea that maybe we could make stem cells, the cells that, you know, we are all formed from before they differentiate into different types of cells in our bodies, into motor neurons. And then we would be able to study motor neurons in a limitless way in a lab setting. And he succeeded in this, and then was part of the team with Kevin Eggen's group at Harvard that made the first stem cell models that actually took skin punches. Now we can also take blood samples from people with ALS and make motor neurons that carry their genetic material in the lab. These are imperfect models, but they really did give us a new window into the disease. And the team at Columbia spent a lot of time thinking about how to use these models in a way that could create a really a new drug screening platform that would recapitulate important aspects of ALS. And that's what eventually led to drug screening efforts and to the creation of Prosetin. I'm going to talk about the platform a little bit because I think it is important. There was a lot of preclinical science that went into the development of Prosetin. This was always developed thinking about what goes wrong in ALS and how can we correct it at the front of our minds. And so I do want to give a little bit of background into how we arrived at Prosetin and at MAP4 kinase as a target. So I mentioned that the team at Columbia was among the first to make motor neurons from people with ALS. But one of the odd things, this was back in 2008, that we found was that motor neurons, if you take skin punches and through a series of applying different chemical reagents, make motor neurons from someone with ALS or from someone without ALS, they don't look that different in the dish. There isn't a huge survival deficit. We sort of expected to see these motor neurons looking really unhealthy initially. And there's been a lot of work done to understand why that isn't the case. But at the baseline, they weren't that useful for drug screening, where you could see an unhealthy ALS motor neuron and then correcting it to something healthy, because they didn't look that sick in a dish. So the first step the Columbia team took was, can we stress these motor neurons in a way that sort of recapitulates aspects of ALS and is selectively toxic to the ALS motor neurons? So we could then drive ALS motor neuron demise in a dish and then actually be able to correct it and see what drugs would make it healthy again. And so they found that compounds that cause endoplasmic reticulum stress, which lead to an accumulation of misfolded proteins, something that we know is a common feature of ALS, were selectively toxic to ALS motor neurons. And then they did the opposite screen, which is how can we rescue ALS motor neurons from ER stress? And they found that there was a set of kinase inhibitors that were really protective to ALS motor neurons in this context. Unfortunately, and because this was an academic effort, we first thought we could just repurpose an existing drug that protects ALS motor neurons in these laboratory settings and use it to treat ALS. But unfortunately, none of the drugs the compounds that were protective in the lab dish were suitable. None of them got into the brain to actually reach ALS motor neurons in vivo. And so the next step was to create a whole set of new compounds, of new chemical entities. The team at Columbia made over 60 analogs and find one that not only protected ALS motor neurons, but also really got into the brain in high levels, so that when it's given to a person, it can actually reach ALS motor neurons even when taken orally. We also spent a lot of time thinking about how this drug works in ALS, because I think we've seen a lot of clinical trials in ALS where the exact target isn't known and it's not able to be measured then in a clinical setting. And so if a trial fails or succeeds, we don't really understand why it's working or not working. And it doesn't give us the ability to learn from that study and sort of build the body of knowledge around ALS drug development. So it was really important to us to understand exactly how Prosetin was working in ALS. And we found through a series of biochemical and genetic experiments that it's actually inhibiting a family of kinases called MAP4 kinases. And this is a complicated slide, but all I'll say is that MAP4 kinases are well established to sort of sit at the top of this cell death pathway that's initiated when motor neurons can't clear misfolded proteins. And we saw that this pathway is blocked. We can stop this cell death pathway if we add Prosetin in a dish. And interestingly, this pathway has also been shown to be activated in postmortem spinal cord samples from people with ALS, including sporadic ALS. So there's not a specific genetic driver that would be meaningful. This is sort of broadly applicable across the disease. We've also seen, really excitingly, I think, in the last few years, several other groups have found that MAP4 kinase inhibition is really neuroprotective in different types of ALS models. And we've also shown that in our ALS models, Prosetin is much more protective than any of the currently approved or once approved standard of care. So really is zolidarbone and sort of the Amylyx AMX0035 combination. So now to the clinical trial. I mentioned we started in healthy volunteers, and this was just to make sure there were no surprises when we gave Prosetin to a person, that we understood its profile and there were no unexpected safety issues. And now we're about to move into the phase 1C part of the study. So the study design for PRO-101, there's a dose escalation portion, which is technically the part C. And this is because the main purpose of the phase 1 study from our view is, first of all, to understand safety and tolerability, also to understand Prosetin's exposure and to make sure that Prosetin is getting into the brain at the levels that we anticipate, to make sure that we have high enough exposure to have a biological effect. And then also to make sure that we're seeing target engagement of MAP4 kinase, so that Prosetin is getting into the brain at the levels we expect and engaging with MAP4K as we've seen it engage in preclinical models. And so we're looking at 3 different dose levels in this study to decide the optimal dose to move forward to a larger future study. This is a fairly small study as planned, 24 participants. There could be more participants if other dose levels are identified to study here, but the main goal here is to choose a dose for further clinical development that is safe and engaging with MAP4 kinase in a meaningful way. The other really important part of this study is that everyone who completes the dose escalating part has the option to roll into a year-long open label extension. And of course, I think this is important because it gives us really good information about how what Prosetin's profile is over the long term. But it also, I think, is obviously important just from the ALS perspective that any clinical trial, I think, should offer an open label extension for people who are volunteering to be part of a development program and give their time and efforts to understanding whether Prosetin is a good drug for ALS. So in the long term, in the open label extension, we'll be able to see long-term safety and tolerability, but we're also going to start to look at exploratory markers to see if Prosetin is having an effect on some of the interesting ideas of ALS disease progression that we can look at in this early stage context. The study is starting in the next month, and we will be recruiting at McGill in less than 4 weeks. So we encourage you, I'll have contact information on the last slide, but please reach out to us if you're interested in joining the study. And we will also be recruiting in Europe at UMC Utrecht. We may be adding a couple of other sites, including US sites later this fall. I just want to mention, I know we're running up against time, but really quickly, because I think it's important. So in a phase 1 study, the main purpose, as I've said, for us is to understand safety, tolerability, and make sure there's a dose that is both safe and engaging with MAP4 kinase appropriately. But that being said, this is the first time that Prosetin will ever be given to someone with ALS, and we want to learn as much as we can from this study to help aid a more rational and streamlined development program, so we can find the right people if there is a specific group of patients that would be more ideal for a later phase study, or a specific timing in the disease, or if there are specific markers that we should really be focusing on in a big way in later phase studies. This is really our opportunity to start to learn this in a meaningful way. So we're looking at, as I mentioned, markers of target engagement to really make sure that Prosetin is behaving as expected in this study, and that there's a dose that is likely to be clinically meaningful. We're also looking at a wide range of ALS-related biomarkers. So things like neurofilament light that are becoming broadly important for drug development. We're also looking at a broad range of markers of inflammation, because we've seen in preclinical studies that Prosetin can reduce inflammation both in cellular models and in animal models of ALS. And we're also looking, in a general way at ALS-related outcomes as exploratory endpoints. We are looking at typical things like the functional rating scale and quality of life. And we're also incorporating some novel AI digital twin approaches, which I think these are unvalidated, but I think if we want to be part of either validating or not validating these technologies, because of course, if we can incorporate some of these AI approaches, it could be really meaningful toward defining subgroups, reducing placebo arms, and other advances in ALS clinical development, not just for this program, but writ large. So we think it's an important thing to incorporate in drug development at this point. And then if this study goes well, our plan is to start a large phase 2 study in 2025. This is a sketch that is not clearly defined at all at this point because we need a lot of data from the phase 1 study and input from all of our advisors and teams to be able to think through what an optimal phase 2 study would be. But the bottom line is that this study will be designed to convincingly address whether Prosetin is actually effective in ALS, and that will be our guiding principle as we move forward. So again, we're about to start recruiting in the next month here. Please contact trials@progenics.com if you'd like more information about the study or if you have any questions. We look forward to answering those questions, and thank you again, Dave and ALS Canada, for this opportunity.
Great. Thank you very much, Erin, for that. That was dead on time. So I don't know if you practiced or anything, but you were right on 15 minutes. So that's amazing, and we really appreciate it. It was a big, long story to go through in a short period of time. I think you did a great job. Yeah, so there have been actually a couple of questions were submitted. And I actually have a couple from myself. One I wanted to start with. So probably a couple of years ago, I saw you give a talk. First time I'd seen anyone present Prosetin, and it was really focused on what you had learned and what had been learned at Project ALS through the landscape of what's important to develop clinical development in ALS. And I know you have a community advisory board at a very early stage. And it was very impressive about how thoughtful you were thinking about the science. And what's really cool is you just delineated that this is a very scientific approach to a phase 1. You're trying to gather as much information as you can to make your clinical development go forward, as opposed to just thinking safety, tolerability, then phase 2, then phase 3. It's like, what can we really get our ducks in a row? But from that community engagement side, could you maybe just expound a little bit more on, Progenics's view of the types of things that you would like to see clinical development in the ALS space specifically? I know you said OLE, but was there anything else that, kind of your vision?
So, to me, and coming from a nonprofit background and then embarking on the early part of this program, really a lot of the very early, there's a lot of bureaucracy involved in drug development that we sort of had to learn on the fly. There's a lot of manufacturing, very boring, but very specific aspects of manufacturing to get from an academic to clinical trial perspective. And the regulatory side of things is also, it's not, there's just a lot of very specialized aspects of drug development that I think we had to learn on the fly again. And what we've seen, with really consistent engagement, we've had the clinical advisory board was expanded, community advisory board was expanded a little bit with ProJenX, but we've had many of the same people advising on this program from the community for years, since day one. And to me, like the education component and having transparent information available about what it means to have a promising target in a laboratory setting, what the next steps are to get it into, some kind of a development. So from discovery to development to actual clinical trials, and a lot of the nuances that come up. I think that maybe in the presentation that you saw, we talked about some of the regulatory hurdles that we faced that we really weren't anticipating. We had a really challenging time. and other companies are in this situation as well, with getting this current study authorized by FDA. And so it just seems like, nothing is happening for 2 years sometimes for companies in drug development. But if we can be more transparent about the real hurdles that we're facing, the real considerations around clinical trials, and then we will be able to get more substantive and real input from our community advisors only if that, it's a two-way street. So I think my general concept is, my general answer is that there has to be a two-way street where companies can't just go to advisory boards, with their set of questions that they want answered. There has to be sort of a back and forth in order for this to be a meaningful relationship. And then specifically, I think the open label, I think considerations around expanded access, which are so different in different regions. Obviously, expanded access is a huge issue in the US, less so potentially in other regions. But really thinking through with our advisors, a humane and regulatorily acceptable and scientifically valid approach to expanded access is something we're thinking about now. And then just getting really practical input on aspects of the study, like lumbar punctures from people who have received lumbar punctures in other clinical trials and have had either very good or very bad experiences, and making sure that we're, putting the appropriate guidance to sites and the people who are conducting the study based on the lived experience that people have had. I think it all comes down to true engagement, though, rather than sort of like a box checking exercise and having community advisors.
That's exactly what I was trying to get at. And I think that that's, I'm a huge ALS nerd myself. I've come from similar, this is many years. And I think one of the unique aspects is that having this company sort of birthed out of a group of people and a discovery that comes from that same community. It's exactly what you just said. It's looking at a partnership with your participants to make this happen. And the way that you've discussed that, I think is very unique and a really nice way to put that out for other companies that are coming into the space, that they should look at their work with the community as a partnership to develop their therapies, as opposed to subjects who
it will be helpful, it will be helpful. Like, I think it's important that companies know if they can engage in this way, it will make their programs better. It will make their studies run better. It will make the data quality that comes out of those studies better.
Yep.
It truly will, it's mutually beneficial to do this. It's not sort of like
Yep. Well, I think it's great. I think it's a great plug for other companies to think more thoughtfully about this, for sure. So you alluded to in there one point where there was the microglial that were reduced, like the sort of not as inflammatory of a signature in mice. And one of the things, there was a question in the chat about whether there was any other evidence of Prosetin value in other models beyond the iPS-derived motor neurons. So obviously it's been given to mice. Did it seem promising as well in other models from different phenotypes or whatever we're seeing?
Yeah, so in the iPS-derived motor neurons, Prosetin has been tested against many different types of ALS stress. And I think also I sort of like blew right through this. But one of the things that has made us really confident in this target, so frankly, we were looking initially for things that just protected motor neurons in general, ALS motor neurons in general. And our development process was target agnostic up until the point that we had something that was brain penetrant, really protective, and we wanted to understand how it was working in order to move it forward. It took us several years to convince ourselves that MAP4 kinase was a good target, because there was no data or other publications suggesting that this was a relevant target in ALS. And it was really around the time that we were coming to Prosetin, there was a group at Genentech that also started publishing data showing that MAP4 kinase was a target of interest in really different models of neurodegeneration, but importantly also in ALS and in Alzheimer's disease. There was a group at Harvard that published similarly in a really different iPS-derived model showing that MAP4 kinase was a good target. And then just this year, or last year, I guess, the preprint, there was a group at UT Southwestern, again, with sort of a target agnostic screen, that found that MAP4 kinase was a really good target in actually directly reprogrammed neurons carrying a range of genetic mutations. They also, in that paper, did test a pretty a MAP4 kinase inhibitor in an SOD1 mouse and saw that it was beneficial. I think our entire approach, so we did test percent, to answer your question directly. We saw improvement in strength deficits and a delay in weight loss in SOD1 mice administered Prosetin. And we tested Prosetin in another ALS animal model, a FUS model that was developed by our collaborator, Neil Schneider at Columbia. And that's where we saw that it could really very meaningfully affect microgliosis or neuroinflammation. I think the whole reason that our team was so involved, or the founding principle of our academic research, was that the mouse models have not typically been that great at predicting clinical success. So in our view, the IPS models, one of the things we'll learn from this development program and from some of the other programs in the clinic now are whether potentially these IPS models are better predictors of clinical efficacy than the mouse models have been in the past. So I think it was important for us to, to be able to confirm benefits in animals, but we are more inclined to celebrate the motor neuron, the IPS-derived motor neuron data.
Yeah, and I think you make an incredible point on the fact that it's also the UT Southwestern group talked about TDP43 specific things that were in there, and then in a G93A SOD1 model where there's not necessarily a TDP43 piece, is it worth studying in that model if you're seeing things that might be effective for a larger group of people who are maybe slightly different? So, I mean, it totally makes sense that given that we don't have great TDP43 models, for example, if that group is correct and MAP4 kinase is a good target for that type of thing, which I would also want to ask, are you curious to see if Prosetin does some of those same things? I noticed also they got Kenpolone out of theirs as well. So that's actually good validation as well for what you saw. But I'm curious as to whether or not you're sneaking around and looking to see if Prosetin's doing some of the other, because they had some interesting nucleocytoplasmic transport, TDP43 nuclear localization. You don't have to tell. You don't have to tell me.
No, we are, of course. So just really, I think very transparently, Progenics is a super small company. I showed we have 2 employees. We don't have any lab space of our own. We still have definitely collaborators at Columbia that are interested in this mechanism. And we found that finding, so not to get too in the weeds in this webinar, but that paper from UT Southwestern proposed an entirely different mechanism for how MAP4 kinase inhibition could be protective than what we have found and what the group at Genentech and the group at Harvard previously published. So I think it's actually very clear to us that MAP4 kinases are doing a bunch of different things, and there probably is more than one mechanism by which they could be acting. We have not recapitulated those studies yet. We are interested in doing it.
Sure.
Yeah,
that's a good point. And so on that note, then one of the questions was around any toxicity because it's a MAP4 kinase, it's high up in a pathway. But I think because you've done these very solid phase 1A, phase 1B steps, you're feeling very confident about that.
We are, And I think also importantly, I alluded to really needing to show a lot of data to FDA around safety in order to get the study up and running. So we've actually also completed chronic tox studies in animals. So we've dosed up to many times higher than we plan to dose in people, over 6 months in rodents and 9 months in non-rodents. And so I think, this was a big concern for us because this is the first brain penetrant MAP4 kinase inhibitor to ever get into the clinic to our knowledge. But we've really become very confident just through the clinical and non-clinical studies that there is a really good safety window and that this drug should be safe and well tolerated at the doses that we're proposing.
Yeah, it's definitely, I mean, it's adding up to be a pretty interesting target for sure. And so last question that came from the chat was around pipeline, because you're a new company, you've just said you're 2 employees. It's a really hard thing, but I guess ambition to a pipeline, is there an ambition to think about other things that maybe come out of some of your colleagues and the individuals who sort of led to Prosetin?
Yeah,
for other
of course.
targets?
And that's one of the, right now, I keep saying we're going to be recruiting in a month. Our sole focus for the next couple of weeks is just getting the study up and running. But one of our other big goals for this year is to start to build our team, build our company, and expand the pipeline. I think importantly, from our perspective, and I think this is probably true for anyone who's honest with themselves about ALS drug development, Prosetin is not going to cure ALS. There's nothing in the clinical, clinic for ALS right now that is a cure for the disease. And so we are really interested in actively evaluating. We have other compounds actually that we licensed from Columbia when we started Progenics that we're evaluating in the background. Other research coming out of our collaborators and beyond that we're evaluating, and certainly bringing in a complementary next program in 2025 is a big priority for us.
Awesome. Well, thank you very much for your time. PRO 101, everyone. I think Montreal should be recruiting fairly soon at the Montreal Neurological Institute. Really appreciate your time, Erin, for doing this, and thanks for joining Clinical Trials Unboxed.
Thanks, Dave. Thanks,
All right.
everyone.
Take care.
Bye.
See you, everyone.